time to start? Okay, great. Just a couple of seconds to ensure that everyone is here with us and we all start at the same time. A couple more seconds. Okay. I still see Sarah here. Let me allow Sarah in. So I think we are all ready. So good afternoon. It's definitely very good to be back after a week's break. I welcome everyone to the Food for Sustainability and Grow webinars, this time dedicated to functional biodiversity. We are thrilled to have you here with us as we delve into a crucial topic that plays a significant role in shaping our food systems as well as sustainable practices. Today, we'll dive into the fundamental principles of functional biodiversity and its pivotal role in shaping sustainable agriculture practices. Our distinguished panel of speakers will bring us today a wealth of knowledge and experience to the table. So you're probably aware of who they are, but first let me introduce you Sofia Costa. Sofia, a renowned nematode ecologist whose research has shed light on the intricate interactions within soil ecosystem. Next up, we have Filipa Tereso, hailing from Agrosustentavel, where she works tirelessly to implement innovative strategies for harnessing the power of biodiversity in agriculture. Filipa, it's also a pleasure to have you here. And last but certainly not least, we have Felix Wackers representing the BiobestGroup, who specialize in developing biocontrol solutions that leverage biodiversity to manage pests in a sustainable way. Together, they will enlighten us in the principles of functional biodiversity and its practical applications in agriculture. Please join me in welcoming our dear speakers. It's really a great pleasure to have you here. And before we proceed, let me remind you some of the house rules. As you'll know, our webinars are recorded, so please just remain with your camera off if you do not wish to appear in the recording. But again, I remind you that we prefer to see you and to see all of you and your faces. So please keep your camera on if you feel comfortable. Also, questions are welcome. You know that we will address your questions to the speakers after their presentations. And for this, we will use Slido or the chat here in the Teams and in a written format. And as I told you before, this is mainly due for time management reasons. So we try to use Slido. Now we are all very familiar with Slido. I believe we all know how it works, but it's really, really simple. Just type www.slido.com in one of your tabs in your browser, or just use your mobile phones. And what you will see is a hashtag box. Just please insert F for S Academy. And once you have done this, and I invite our speakers also to join our Slido to test your knowledge, and we will use these questions as usual for a small warm-up to the topic that we have here at hand. So are we ready? And Rita, let me know if we are ready. Okay, so you can go to the Tools tab. Okay, and the first question relates to a very particular animals on Earth, the nematodes. Do you have an idea of the proportion of all animals on Earth that nematodes account for? 20%, 40%, or 80%? So 1/5 to fifth or fifth. And while you take your time to consider these figures, I will ask Sofia to tell us a little bit more about the geographical dispersion of nematodes throughout the globe, because I found these findings very counterintuitive. And it would be quite interesting also to understand the rationale behind this. Also, quite interesting is to know the size of nematodes. They can range from a fraction of a millimeter, so very tiny, tiny, tiny animals, to actually, I don't know, Sofia, correct me if I'm wrong, but a few meters. I've heard that the nematodes that live in the gut of the cachalot waves can be fairly big. So hopefully, Sofia, you can then let us know a little bit more throughout your topic about these numbers. I found this curiosity is very interesting. But let me go back now to our question. Yes, indeed, you're right. You're always a very knowledgeable audience. Thank you. Indeed, is 4/5, 80% of nematodes accounts for the total animals on Earth. So we as the human beings need to be very careful and treat them well, because if they get mad, there are quite a lot of them, so we need to be careful. Thank you so much. So we will start with nematodes, and then Peter, can we move to the next question? This is about functional biodiversity, which is intrinsic linked to food webs. So, for example, how can you effectively control a slug or snail invasion in your vegetable orchard? You could control this by introducing ducks, by using biopesticides, or, for example, by installing physical biofunctional barriers. We have a tie here. Well, I could say that we could invite all the snail lovers and the good chef, and surely this would solve the problem. Just by the way, snails are a much appreciated delicatessen, mainly in the south of Portugal. So if you visit Portugal, do not miss this. But while we cannot invite all the snail lovers, what could it be done? Okay. So again, we have a clear winner. And yes, you're absolutely right. Ducks can help, can assist in solving the problem. Filipa, if I'm wrong, your assistance here, please, also, because let's hear how the ducks work. But it could be a way to work along the lines of functional biodiversity and the food web. Great. So ducks in our minds. And next we revert to pollinators. Pollinators is Felix's passion. So pollinators are one very important functional group for ecosystems. Do you have an idea what percentage of flowers, including fruit and vegetables, are pollinated by bees? I think we all have a good understanding that bees are great pollinators, and they represent a very important role in the function and the pollination of flowers. They've been, covered a lot in the media and raised awareness campaigns about protecting bees. But do you have an idea? Well, I think here it's quite straightforward. I think everyone knows that around 75% of flowers are pollinated by bees. Felix, let us know more about bees and how we can use them in our favor. Thank you so much. It was great. Again, congratulations our audience for being so well prepared into our topic. So while I call Sofia to the floor, let me just briefly introduce you to Sofia. So Sofia is a nematode ecologist based at the Center of Molecular and Environmental Biology at University of Minho. Sofia has a PhD from Lancaster University and a post-graduation in organic agriculture. Sofia's research interests follow two main lines of research. First, the use of nematode communities as bioindicators of soil-based ecosystem services, and secondly, the ecology and control of crop-damaging plant parasitic nematodes in agroecosystems. Sofia, it's a pleasure to have you here. Thank you so much. You have your presentation, so the floor is now yours. Thank you.
Thank you, Claudia, and thank you for inviting me to give this talk in in your series of webinars. I understand that Food for Sustainability is hosting an online course on soils. So this is sort of related possibly to that course. I'll be telling you about nematodes, not in such generalistic terms as we were talking about earlier. So I'm not mentioning the really long, 9 meters long ones that parasitize whales, but I'll be telling you about nematodes that live in soil and how we can use their functional biodiversity to provide indicators of the status, the ecological status of soil, and we can use that to then support sustainable soil management in agriculture. So I'll try to be brief and try to keep it to 15 minutes, but I'd like to give you an overview of how we can use nematode community analysis to do that, and give you a couple of examples of work that we've done in our group, which is a nematology group at the Center of Molecular and Environmental Biology here at the University of Minho. So to start in broad terms, we're looking at soils, and soils, of course, is where food begins. And soils have been forever used as just, or interpreted as, perceived as scaffolding, where plants grow and allow them to root. So usually people think of soils as an inert structure of mineral contents, of maybe organic matter, water and air. And they forget sometimes that soils are actually alive. And there's lots of living organisms in soil. Soil is a habitat, and it provides us a lot of different functions. I've listed them here in fairly broad terms as well. So healthy soils provide water, they contribute to the water balance, they can absorb water and detoxify chemicals in it. Soils recycle nutrients, so they're involved in all these biogeochemical cycles that end up having nutrients for the plants. They store carbon. Soils are very important for carbon sequestration. So all the carbon that's not in the atmosphere is actually good, because carbon, like CO2, is a greenhouse gas. So we want to keep carbon, inert in soils, stored in organic matter. And healthy soils, through the living organisms, also regulate pests and diseases. And it's something that we've been working on for quite a while. So there's all these functions, and of course, among all functions, those that benefit us, humans, are called ecosystem services. They're sort of nature's contributions to people. And in agriculture, you have this graph here showing you the levels of different ecosystem services, and I've got pollination there as well, so for a sort of introduction for Felix as well. There's pest regulation, pollination, availability of nutrients, and a good water source as well. These are all services that soils among the full ecosystem provide. And what's being given by the ecosystem is something that the farmer then doesn't have to intervene. So if you've got pest regulation by your ecosystem, by your organisms, you don't have to apply as many pesticides or think of clever ways to try to regulate pests and diseases. Of course, these ecosystem services are provided by biodiversity. And in soils, we have a multitude of organisms. And of course, all of us possibly have already seen moles and little crustaceans there, and ants and earthworms, but you have to start looking very carefully if you want to find these sort of minor insects and arthropods. And then if you have a microscope, then you can pick up on other organisms, including nematodes there. But of course, in living soil, in a very small volume of soil, you have millions of bacterial cells and hyphae, you've got mycorrhizae, you've got all sorts of organisms. Now, among them, you've got the nematodes that I'll be telling you about. Nematodes are animals. It's something that sometimes we don't think about. They're very simple animals. We've got them there. Most of them, the nematodes in soil, are all microscopic, transparent, tiny little worms that are buried in soil, so not many people are aware of them. And in fact, they're the most abundant animal. And you've got here this huge number of soils, of nematodes only in the first 10 centimeters of topsoil. And then you've got nematodes in deeper layers of soil and in water sediments and mostly anywhere really. And they're not all the same. You know, you've got here, I've got listed nematodes according to what they eat. So they're trophic groups, they're feeding groups of nematodes. You've got nematodes that feed on bacteria, feeding on fungi, feeding on plants, and you've got plant parasitic nematodes there that farmers sometimes struggle with. And then you've got omnivores and predators. So you've got all the levels in the food web of the soil. You've got nematodes representing them, except of course for the producers. The producers are the plants. And if you start with the plant roots and you start following, and this is a food web that I'm only illustrating, or mostly illustrating nematodes here, but you've got plant parasitic nematodes, but then there's a parallel food chain in soil, not starting in plants, but actually on organic matter that's being decomposed by bacteria and fungi. And then you've got bacterial and fungal feeding nematodes feeding on those organisms. And then if you keep going, then you've got predatory nematodes. And this is just to say, if you have a good idea of how the nematodes are doing, what trophic groups, what life strategies.
Sofia, sorry to interrupt. Are you moving the slides?
Are you stuck in the first?
Yes, we are. And thanks for the note in the chat. I thought it was on me.
Okay, because I can't see the chat. Hang on. So what if I do this? So this is the first slide. Are you seeing it?
Sofia, I would suggest that you switch off your camera because apparently your image is kind of froze also. Okay. Let's see if we can improve connection like this.
All right, I'll switch off the camera and then I'll go through everything again. So now you know what I'm going to say. So that's very unfortunate. Okay, I'll start screen sharing again. And I'll share. So I've shared the entire screen previously. Now I'll share my presentation. And it's a different, hang on. So can you see the first slide?
Well, not from my end. It's probably Rita,
no. I can't see your slides.
Just tested this. Right, we just tested this and it was fine, and now it's not working.
Exactly.
Okay, so I don't know what I'm doing. Can you see the first slide now?
No, we can't see anything, Sofia.
All right, but so I'll stop sharing that. And I'll...
Well, Sofia, the alternative is for Rita to share from your end. Rita, you have Sofia's presentation?
I don't know.
Do you?
I don't think so.
But is it there now? No. No. Okay.
Yes, it's blank. It's not coming. It's probably internet connection, Sofia. The only thing I can offer for you to send Rita's the presentation. Rita will share.
All right. And you can move on to the next one, and I'll.
Yes.
All right. Okay. I'll do that.
Well, apologies for that. We always test everything before we start running the webinars, but then sometimes Teams has its own personality. We're really following. It was very clear in terms of your speech. It came through very clearly, but unfortunately the presentation didn't come as good. So if we can...
sorry about that. Not sure what happened there, but okay. I'll send vou mandar a Rita.
And I will share.
Okay, sure. So please bear with us a couple of more seconds. Things happen, but surely we're very keen to continue to know more about nematodes. I wasn't aware that there could be a 9-meter nematode, which is Pretty scary. Okay, still some participants joining. Okay. Alternatively, if it takes long, we can ask Filipa to,
Yeah.
what do you think?
Sim,
Sofia?
por mim,
Yeah,
podemos
okay.
avançar. Sim.
Okay, good. So while Filipa prepares to come onto the stage, so Filipa is going to speak in Portuguese, and she will have the presentation in English. So not worry, and I think you can try some functions on the chat on Teams. We just try that, and you can have the translation in English. You know, quality and credits given to Teams. So, but we'll try to translate when we make it live. So a brief introduction about Filipa. So Filipa holds a master in conservation biology from the University of Évora, and Filipa has over 20 years experience in olive growing and viticulture. Filipa began her career at AJAP, which is the Portuguese Young Farmers Association, and in 2007 she became a manager at Esporão. Currently at Agrosustentavel, Filipa performs technical work in the area of biodiversity, sustainability, and organic farming. Filipa, thanks for jumping to the floor in such a rush. It's a pleasure to have you here, and we look forward to hear your presentation.
Obrigada. Estão a ver a apresentação, só para confirmar?
Yes, all is going well. Thank you.
Então, antes de mais agradecer o convite, de facto é uma honra estar aqui a falar de um tema tão importante como é o da biodiversidade, ainda mais fazer aqui esta ligação da biodiversidade com as explorações agrícolas. De facto, quando nós falamos de biodiversidade, no fundo estamos a falar de diversidade. Diversidade genética, diversidade de fauna, diversidade de flora, mas também diversidade de ecossistemas. E dentro dos ecossistemas, obviamente que nós encontramos o ecossistema agrícola, que tem as suas particularidades, porque não é um ecossistema natural como os outros, é um ecossistema que tem, de facto, intervenção humana e, portanto, dá-lhe características muito próprias e que devem devem-se gerir de forma de um ecossistema e não só de uma cultura. E nesta situação, quando nós estamos perante um ecossistema agrícola, há a componente da biodiversidade que é muito importante. E porquê? Porque se nós queremos trabalhar para um equilíbrio biológico, a biodiversidade é a base. Não é possível atingir um equilíbrio biológico se não tivermos biodiversidade na nossa exploração, na nossa cultura. Esta biodiversidade, para além de permitir este equilíbrio biológico, que vai ser importante depois também para a gestão das pragas, é importante porque nos confere uma resiliência muito grande à componente da cultura. Ou seja, se estivermos perante uma cultura que está rodeada de biodiversidade, o que nós estamos a fazer é que ela tenha capacidade de ter respostas positivas mais rápido e mais eficientes a qualquer impacto negativo que tenhamos e que, nomeadamente, por exemplo, as condições climáticas. Portanto, há que trabalhar no sentido de atribuir uma resiliência maior a este ecossistema agrícola. Quando trabalhamos com este objetivo, nós vamos ter possibilidade de usufruir dos chamados serviços de ecossistema. Portanto, a biodiversidade, no fundo, será o início que nós temos que trabalhar para podermos usufruir, então, de serviços que nos são dados de uma forma gratuita pela natureza, se soubermos, assim, trabalhar com ela. E como exemplos destes serviços de ecossistema, nós podemos referenciar alguns, eles são muitos, mas alguns, como o sequestro de carbono, a regulação de polinizadores, a regulação da fauna auxiliar, a fertilização dos solos e a regulação climática. Portanto, são alguns pontos e alguns serviços de ecossistema que, de facto, são importantes para o agricultor e para a gestão, não da exploração agrícola, mas sim do ecossistema agrícola. E, no fundo, o dilema que se coloca para o agricultor é se vai continuar a trabalhar numa exploração, onde temos única e simplesmente a cultura e o objetivo principal é a produção, ou passamos a olhar para a nossa exploração como um ecossistema e neste ecossistema vamos ter que decidir também que tipo de agricultura é que vamos praticar. Se praticamos uma agricultura que é só consumidora dos serviços do ecossistema e, portanto, neste caso, o caminho será o desequilíbrio deste ecossistema e, portanto, nunca iremos atingir a tal resiliência que é necessária, ou, pelo contrário, optamos por uma agricultura que é potenciadora desses serviços e, neste caso, estamos a trabalhar com a natureza, estamos a ter aportes dos serviços do ecossistema e, portanto, trabalhamos para a estabilidade do ecossistema. Se a opção for esta última, obviamente que, mais uma vez, a biodiversidade é o ponto fulcral. E porquê? Porque dentro desta biodiversidade nós podemos ir buscar ferramentas que, no fundo, podemos utilizar na gestão das nossas explorações, nomeadamente a biodiversidade funcional. No fundo, esta biodiversidade funcional é uma componente da biodiversidade que tem uma função imediata para o agricultor. No fundo, é um serviço do ecossistema e que se baseia, sobretudo, na regulação das pragas das nossas culturas, através de uma ação muito sólida da fauna auxiliar. Portanto, estamos a falar, no fundo, de um controle biológico. E não estamos a inventar nada, portanto, este conceito de auxiliar já existe há muitos anos. Portanto, eu meti-vos aqui um exemplo das cartilhas do lavrador, de 1930, e que já nesta altura fez uma edição única e exclusivamente sobre auxiliares, com este termo, auxiliares, e fazia uma descrição já dos ciclos de vida dos auxiliares, fazia uma descrição dos tipos de auxiliares que temos, portanto, parasitoides, parasitas, as diferentes formas de ação de cada um destes insetos, e também de algo que é muito importante, que era o que é que era preciso existir nas nossas explorações para que estes auxiliares, de facto, tenham eficácia e sejam eficientes na sua ação. E o que é preciso existir é isto, são infraestruturas ecológicas. Portanto, no fundo, são áreas que existem nas explorações para além da cultura e que, no fundo, são o que vai conferir a resistência da nossa cultura a pragas e doenças. Estas infraestruturas ecológicas têm alguns critérios, portanto, elas terão que existir num espaço até 150 metros da nossa exploração, da nossa cultura, para que também tenham um valor ecológico, ou seja, para que tenham uma ação benéfica para a nossa cultura. E, neste respeito, a OILB, que é a Organização Internacional de Luta Biológica, é muito concreta naquilo que diz, ou seja, as infraestruturas ecológicas, para estabelecerem a fauna auxiliar, elas precisam de representar 5 a 10% da área da exploração. Quando nós pensamos nestas áreas, em algumas situações, pode ser difícil perceber aonde é que vamos buscar e aonde é que vamos colocar estas infraestruturas ecológicas. Mas, de facto, se olharmos para o coberto vegetal, se calhar não será assim tão difícil. O coberto vegetal, da forma como é instalado nas nossas culturas, principalmente em culturas permanentes, a forma como o agricultor também já tem uma facilidade em trabalhar com esta estrutura, a maneira como nós podemos enriquecer este coberto vegetal em termos de espécies florísticas, torna esta unidade não só num coberto vegetal, mas num corredor ecológico. Portanto, tem um valor ecológico adicional. No fundo, estes corredores ecológicos são faixas de vegetação que vão unir zonas de conservação ou zonas para além da cultura e que estão separadas e que permitem a movimentação da fauna auxiliar. E muitas vezes o problema da eficiência da fauna auxiliar é nós não termos a coincidência dos locais onde aparecem as pragas com o aparecimento da fauna auxiliar, porque quando estes cobertos vegetais não existem, ela não consegue movimentar-se para o interior da parcela. E, portanto, a base da introdução de biodiversidade nas explorações deverá sempre começar com o enriquecimento e o estudo do coberto vegetal. Obviamente que ele sozinho não funciona ou não tem a mesma eficácia. E porquê? Porque nenhuma destas técnicas vai evitar as práticas culturais que iremos fazer sempre nas nossas culturas. Sejam a passagem de um trator, seja as pessoas a trabalhar, seja eventualmente algum tratamento, seja de fertilização ou mesmo tratamento químico. E, portanto, nós, na nossa gestão da exploração, vamos ter sempre alturas em que vamos diminuir vamos ter um impacto na população de fauna auxiliar. E, portanto, para que haja um funcionamento mais eficiente, nós temos que adicionar, para além do coberto vegetal, áreas de compensação ecológica. E porquê? Porque estas áreas de compensação ecológica vão ser colocadas em zonas estratégicas da exploração, suficientemente perto das culturas, de forma a que criem uma ligação com o coberto vegetal e permitam fazer a recolonização da fauna auxiliar sempre que ela tem um impacto negativo dentro da cultura. E, no fundo, estas áreas de compensação ecológica vão servir de reservatórios de biodiversidade e vão ser a fonte principal da fauna auxiliar. Portanto, vão permitir que esta fauna auxiliar se estabeleça e resida na exploração de uma forma mais homogénea durante todo o ano. Nós temos aqui vários exemplos, portanto, estas SEBs ou estas infraestruturas ecológicas podem ser de vários tipos. Podemos ter vários estratos herbáceos, estratos arbóreos, estratos arbustivos, sendo que o ideal sempre será construir SEBs estratificadas, ou seja, com vários níveis de crescimento para facilitar todo o movimento da fauna auxiliar dentro das SEBs e das SEBs para as nossas culturas, mas também podem ser outras estruturas que tenhamos em que não é possível colocar cultura e que, de facto, possam ser enriquecidas com outras espécies e feita uma manutenção que vá de encontro à preservação da fauna auxiliar. E aqui, nestas situações que eu vos estou a mostrar, os muretes, os pequenos lagos, algumas ilhas dentro de culturas intensivas, não potenciam só o aparecimento de fauna auxiliar inseto, mas também outro tipo de fauna auxiliar importante para o controle de algumas pragas, nomeadamente répteis, pássaros, etc. Obviamente que estas infraestruturas, seja o coberto vegetal, seja outras SEBs ou outras áreas de compensação ecológica que existam, a ideia é sempre que exista uma riqueza específica diversa, maior, ou seja, quanto maior for a diversidade de flores, maior é a diversidade de insetos que eu vou ter. A questão aqui é o balanço, ou seja, é importante perceber que para a biodiversidade funcional, ou seja, para trabalharmos com a fauna auxiliar, o ideal será colocar entre 10 a 20 espécies de famílias diferentes nestas estruturas ecológicas, porque também quando fazemos uma diversidade muito intensiva de diferentes espécies, o que nós podemos estar a potenciar é um aumento também de alguns fitófagos, para além das pragas normais dessas culturas. E, portanto, na seleção de espécies, interessa a diversidade, mas é sempre a diversidade com alguma contenção e ter alguns cuidados na escolha destas espécies, quer em número, quer nas famílias que se escolhem. É importante também fugir um bocadinho às espécies que sejam da mesma família do que a cultura que exista na exploração. Outras questões importantes passa pela adaptação das infraestruturas ecológicas aos insetos auxiliares. Portanto, nós temos características muito bem definidas e muito próprias dos insetos auxiliares, nomeadamente nas suas características morfológicas e do seu aparelho bucal em específico. Estão aí 4 tipos de aparelho bucal específico e isto significa que para alimentar na componente açucarada estes insetos, as flores têm que estar adaptadas ao aparelho bucal e, portanto, quando nós fazemos a seleção de espécies a introduzir, temos que saber previamente que ordens de auxiliares é que nós queremos aumentar. E para isso precisamos de saber quais são as pragas principais, quais são os auxiliares que maior eficácia têm no controle dessas pragas e depois então partir para a seleção das espécies florísticas a introduzir, quer no coberto vegetal, quer nas áreas de compensação ecológica. A outra questão a terem atenção e que também é muito importante para que esta ferramenta de facto funcione é a distância a que nós colocamos estas infraestruturas. E aí a importância ainda maior do coberto vegetal porque vai estar dentro da cultura. Estes insetos que nós estamos a falar, muitos deles são pequenos, não se vêem sequer sem o auxílio de uma lupa e, portanto, a sua forma de movimentação é também muito diferente. Nós temos auxiliares que se movem até aos 10 metros, 50 metros, e temos outros que têm uma capacidade de voo maior e que podem ir para mais de 500 metros. Ora, obviamente que, conhecendo os auxiliares que queremos aumentar, nós temos que adaptar os sítios aonde vamos colocar estas infraestruturas, porque se colocarmos muito longe, por exemplo, espécies que são indicadas para aumentar parasitoides, que têm capacidades de movimentação mais pequenas, nós nunca vamos ter esses parasitoides na cultura se as sebes se localizarem muito longe da parcela. Portanto, a adaptação das infraestruturas ecológicas para que, de facto, elas tenham um valor ecológico e desempenhem esta função, nós temos que ter alguns cuidados na sua introdução, no seu melhoramento. Quando trabalhamos com estas questões, obviamente que isto é uma ferramenta, e é uma ferramenta útil para o agricultor, porque conseguimos perceber, ou tentamos pelo menos, ter mais certezas na escolha das épocas de tratamentos, se de facto é preciso tratamento ou não, e a ideia aqui é reduzir o número de tratamentos que se faz. E com este conhecimento nós conseguimos ter uma segurança maior nessa decisão. Nós conseguimos perceber o que é que nós temos nas nossas explorações em termos de abundância, fazer essa relação da abundância, ou seja, da quantidade de indivíduos que temos com a riqueza específica, ou seja, que quantidade de espécies é que eu tenho da mesma ordem, e o que interessa é sempre diversificar, ou seja, ter mais espécies do que propriamente o mesmo número de indivíduos, portanto, ter mais diversidade de espécies. É importante perceber a relação que nós vamos ter entre a fauna auxiliar e os fitófagos. Portanto, eu pus-vos aqui alguns exemplos em que vocês têm uma percentagem de fitófagos de fauna auxiliar que foi encontrada. É muito diferente quando nós afunilamos e percebemos que dos 34% que tenho a representar os fitófagos, só 26% desse valor é que de facto são fitófagos problemáticos para a minha cultura. A mesma coisa relativamente à fauna auxiliar. Toda ela é importante, mas é bom saber que de toda aquela que é identificada, a maior parte corresponde àqueles insetos que têm uma ação mais eficaz no controle das pragas da minha cultura. E, portanto, tendo estes resultados, permite-nos, de facto, ter aqui uma gestão mais apropriada, mais focada das nossas explorações. Conseguimos também perceber algo que é muito importante e que nos permite perceber até que ponto é que esta ferramenta está a funcionar, que é o estudo das dinâmicas das populações. Tenho aqui um exemplo de uma vinha que desde o início está em agricultura biológica e por isso é que também temos estes resultados, mas é fácil perceber que aqui já existe uma dinâmica que é acompanhada, ou seja, a dinâmica da praga é acompanhada pela dinâmica da fauna auxiliar, e isto faz com que eu tenha a certeza que eles vão estar na mesma altura e vão se encontrar e, portanto, a eficiência da fauna auxiliar vai ser maior. É também perceptível neste caso que eu tenho já um valor de fauna auxiliar, no caso da vinha, superior à população da praga. Isto é indicativo de que o que está a acontecer nas sebes está a passar para dentro da cultura. É este o objetivo quando nós fazemos uma ligação de espécies entre o que está em área de compensação ecológica e o que está depois nos cobertos vegetais, e conseguimos perceber que existe já uma evolução do número de espécies de fauna auxiliar relativamente à praga. E, portanto, claro que o ideal será o valor ser superior, portanto, passar-se um bocadinho mais ao que nós temos aqui nas sebes, mas já é indicativo de que a existência de diversidade em termos de coberto vegetal e de infraestruturas ecológicas é, de facto, uma mais-valia. E para terminar e não passar muito aqui o meu tempo, deixar-vos também aqui algo que eu acho que é muito importante e que vem muito associado a esta questão da biodiversidade, que é não só a parte técnica que é útil para o agricultor e que tem um impacto de facto na gestão da exploração, mas depois também no que se pode ir buscar quando se trabalha com a natureza. Portanto, a questão da biodiversidade faz com que hajam outros serviços que são importantes pensar, nomeadamente relativamente à parte turística, e eu meti aí o Portugal Farm Experience, que tem um leque já de empresas que trabalham com esta questão da valorização da exploração através do trabalho que se faz da biodiversidade das explorações. Obrigada.
Filipa, thank you very much. It was a very, very interesting and enjoyable presentation. Thank you for your time, and hopefully soon we'll also be hearing about biodiversity credits to remunerate those that really invest in biodiversity. While we wait for the questions in slide or in the chat, I have here two questions for you. So you clearly mentioned the crop and productivity and the challenges that some farmers face, that they might consider that they are taking out part of the farm that should be addressed to crop and to crop yield. Do you have any idea from your experience what are the trade-offs so that farmers can be more encouraged to actually do these hedges and contribute to cover crops to enhance biodiversity?
Cláudia, pode-me dizer em português
Claro
que eu
sim,
percebi
claro que
parte da
sim,
questão?
desculpe, Filipa. Claro que sim. Pronto, o Filipa abordou na sua apresentação muito bem que há sempre aqui um risco percebido entre produção e área alocada às questões da biodiversidade. Filipa, perguntava se reforçar um bocadinho mais da sua experiência, quais é que são aqui os trade-offs? Nós, de facto, perdemos, ao perder a área, perdemos, de facto, a área produtiva, mas se calhar ganhamos por outro lado. Qual é a experiência da Filipa e em termos de percentagens nós estamos a falar?
Eu nunca não associo a perda de produção a estas áreas de compensação ecológica. E porquê? Porque normalmente estas áreas de compensação ecológica existem nas explorações, só não são aproveitadas como tal. Portanto, há zonas de exploração que não têm cultura porque sabiam que ou o solo não estava adaptado, ou tinha muita pedra, ou tinha uma zona de árvores que não arrancaram, e portanto não é retirar cultura para colocar estas áreas de compensação ecológica, é sim valorizar o que não é produtivo e dar-lhe valor ecológico de forma que trabalhe numa sintonia com a cultura. Portanto, não se trata de diminuir produção, trata-se sim de valorizar áreas que não tinham valor ecológico atribuído. E aqui está uma mais-valia muito grande para o agricultor, porque de facto mantém a mesma área, mas está a utilizar algo que não lhe dava dinheiro e que lhe vai prestar um serviço de ecossistema e que vai ter utilidade depois na regulação de pragas.
Interessante. Filipe, eu tenho só mais uma questão antes de passar aqui aos nossos participantes, e eles é que são a estrela e não eu, mas só uma curiosidade. Filipe abordou as questões do coberto vegetal, não é? E o coberto vegetal a ser trabalhado e escolhido por forma a promover determinado tipo de espécies, que são as espécies benéficas e aquelas que podem combater algumas pestes. Nós aqui no Food for Sustainability trabalhamos bastante com o microbioma do solo, portanto a biodiversidade, outro tipo de biodiversidade, e fazemos a recomendação destes cobertos vegetais com base, portanto, nas comunidades de micro-organismos que foram identificados. Vocês, nesta identificação do coberto vegetal, têm em consideração alguma parte do microbioma do solo? Ou ainda são duas áreas independentes?
Podem ser duas áreas independentes ou não. Portanto, quando é escolhido o coberto vegetal, e aqui não tem propriamente só a ver com a biodiversidade funcional, mas quando se escolhe um coberto vegetal para se instalar numa exploração, deve-se pensar na funcionalidade desse coberto vegetal. Para que é que eu vou instalar o coberto vegetal? Se é para ter um impacto no solo em termos de estrutura, de reduzir compactação, aumentar infiltração, melhorar a microbiologia do solo, eu vou escolher espécies que vão ao encontro dessas situações. Pensar dentro dessas espécies, que outras espécies é que eu posso introduzir que sejam mais ricas em termos de florísticos e que eu possa introduzir também que tenham um aporte para a biodiversidade funcional, sabendo logo à partida que algumas dessas espécies vão ter essas funcionalidades todas. Portanto, quando nós escolhemos ou quando nós selecionamos, é possível selecionar para aumentar a diversidade que está no solo, até porque isso é importante, aquilo que referenciou, porque muitas destas infraestruturas não têm um impacto só na biodiversidade funcional em termos de insetos da parte aérea, têm um impacto também do aumento do microbioma do solo, sempre, não é? E, portanto, essa conjugação pode ser feita e esses benefícios podem andar em conjunto. Nós podemos escolher espécies florísticas que vão ao encontro desses dois objetivos.
Excelente, Filipa, muito obrigada por este esclarecimento e continuamos a trabalhar isto de facto, está tudo ligado. Duas perguntas aqui no chat, brevemente, Filipa. Alguns trabalhos na cultura do arroz que vocês tenham desenvolvido?
Não, na cultura do arroz não, mas sei que não é tão fácil porque não é uma cultura como uma cultura permanente, por exemplo, mas é uma cultura que tem uma taxa de ocupação de solo bastante grande, não é? E, portanto, na cultura do arroz funciona muito bem tudo o que seja infraestruturas anexas à cultura. Portanto, o reforço da biodiversidade deverá ser em anexo à cultura. E aqui, como se trata de uma planta que no fundo é herbácea, o cuidado maior que terá de haver é de facto na escolha das espécies que se vão introduzir. Fugir das mesmas famílias, porque a probabilidade de se aumentar o número de pragas nesta cultura pode ser maior do que um trabalho destes para uma cultura permanente.
Obrigada, Filipa. Uma última questão. Falamos aqui durante a apresentação de 5 a 10% da área dedicada à biodiversidade funcional. Porquê 5? Porquê 10? Porque não mais, menos?
Os 5 ou 10% é um valor que é indicativo pelo OILB, pela Organização Internacional de Luta Biológica. Portanto, para além de definir os 150 metros dos estudos que eles fizeram, definiram também que o impacto que estas infraestruturas ecológicas têm para estabelecer a fauna auxiliar, ou seja, o estabelecer da fauna auxiliar não é encontrarmos periodicamente fauna auxiliar nas explorações, é de facto conseguirmos encontrá-la durante o ano inteiro, nos vários estágios do seu desenvolvimento. E para que isto de facto aconteça, dos estudos que eles fizeram, estes 5 a 10% são os necessários de biodiversidade, ou seja, para além da nossa cultura, que outras espécies é que poderemos colocar dentro desta percentagem.
Obrigada, Filipa. Thank you so much, Filipa.
Nada,
We now
obrigada eu.
need to move on. Thank you so much. Please stay with us. Sofia, I now invite you to continue your presentation. Rita, would you be too kind to share Sofia's presentation? Sofia, can you hear us?
Yeah, I can. Can you hear me?
Yes, fantastic.
Okay.
Okay.
Good.
Sofia, thank you. So we were stopped in the first slide. We were knowing a little about nematodes and who they are and what they look like. So maybe we can resume from there.
Yeah. Fine. Well, thank you, and sorry about that glitch. So here I was telling you about how soils are not just structure for the plants to root. They have several functions, and I was telling you about how these functions, if they're, I mean, now Filipa has talked about ecosystem services as well, but some soil functions are directly important and beneficial to us humans. And in agriculture, anything that can promote nutrient recycling, nutrient retention, water detoxification, help combat pests and diseases, it's something that's very important, because then farmers don't have to themselves resort to chemical inputs or any other inputs that they bring into the system. So click, Rita, if you click. Thank you. Okay, it's good to see them moving now, isn't it? So all these ecosystem services based in soil are provided by the biodiversity of soil. And you've got millions of bacteria and fungi and all sorts of different organisms of different sizes. And the moles that you see there on the bottom right corner are actually the megafauna. So that tells you that, you know, mega, huge fauna is going to be a small mole, because all these organisms are really very tiny. And among them, you've got the nematodes. So if you click, Rita, nematodes that you see there, there's a couple of pictures there on the right. You see nematodes, there are those worms there. They're microscopic, transparent worms that live in soil. They're hugely diverse. They're the most abundant animals, and they're the animals that are more diverse as well. And not just taxonomically, but also in terms of their function. So this was an exercise, that map there, that's mapping the soil nematodes across the globe. And it's based on estimates on samples, a huge sampling effort from several nematologists across the globe. And not only did we quantify them, we actually categorized them also according to their feeding habits, so their trophic group. You've got bacterivores and fungivores and herbivores, omnivores, predators, all these different types of nematodes That's functional biodiversity. You can categorize nematodes on trophic groups and in life strategies, and you can come up with functional guilds. So if you advance the slide, and we can see the food web in the soil, can we? Yes, we can now. So now we've got a very simplified food web, and it's only depicting nematodes. So I've sort of, well, there's plants and bacteria and fungi there as well, but I got rid of all those other organisms that you saw in the previous slide to make it more simple. But this is just to illustrate that if you start on the left with the producers that are the plants, then you've got plant parasitic nematodes that are herbivores that feed on those roots. And they can be very difficult to manage in agricultural systems where herbivores are feeding on the crops, you know, that you want to produce, that you want to get yield from. Now, there's a parallel food chain there that's based on organic matter. Anything that used to be alive and is now not is going to be decomposed by bacteria and fungi. And those decomposers are eaten then by bacterial and fungal feeding nematodes. So they're on the third trophic level. So if you continue on the food chains, you'll reach predatory nematodes. So as you can see, nematodes are represented in all of the trophic levels in the food chain, except the energy source, the producers. And if you have sort of fingerprint, an image of what all these different groups of nematodes are doing in the food web, then you know a lot of all the processes that are taking place in the food web, in the soil. So just as a take-home message there, soil nematodes are taxonomically and functionally diverse. They're absolutely everywhere in big numbers. They're present in all biomes, in all types of soil, and they're all in all trophic levels in food webs, and they're involved in all those processes that are taking place in the soil. So they're used as bioindicators. They're not used as bioindicators in the sort of, in that essence of being sentinels of, or being biosensors. But if you've got a sense of their whole community, if you've got a tool to analyze their communities, then you learn a lot about the soil. So next slide, please. Oh, that's okay. So you've got, oh, go back. Sorry. Yeah, there's the animations there that I wasn't taking into account. So yeah, you've got three key processes here. Herbivory is, of course, a disservice. It's a negative impact in our agricultural systems. Of course, in natural systems, it's not a disservice at all, but we can discuss that later. And then you've got them involved in decomposition and regulation. So regulation of pests and diseases and of the full trophic, food web. So if you click now, Rita, thank you. So how can you then summarize your full nematode community into one data point in the graph that's there on the left? On the y-axis, you've got nutrient enrichment, so how much nutrients are available to the system. And on the x-axis, you've got a crescendum of structure, of food web complexity and stability. And if you've got all these indices that we can calculate from nematodes, and we can summarize the full community to one data point in that plot. And according to what quadrant it's going to land in, then you know if your soil is enriched in nutrients, if it's being regulated, if it's got the conditions to become suppressive to pests and diseases. And that sort of information can be used to then act on the soil. How do you want to manage your soil to try to bring it to the fertile and suppressive conditions that you would want in agriculture? So if you click again, there's going to be another thing coming up on the slide. Yeah, there it is. So if you then take into account all the biomass and activity that's standardized, and we can calculate very easily for the nematode communities, you then come up with metabolic footprints of each of the nematode different groups and the structure and the enrichment indices. So you can sort of target where the nutrients are allocated in the full food web. And this decomposition, herbivory, and regulation functions as inferred from the nematode community analysis are actually very direct indicators of those functions in the soil. So if you advance the slide, so we've been using this sort of analysis to try to come up with a good indication of where we're going when we're applying different practices in soil, or comparing different soils from different land uses or different agricultural management. And of course, everyone's trying to come up with better, more sustainable solutions to make the system resilient and to naturally improve soils and the full ecosystem. Of course, soils are a component of the ecosystem that's been neglected for a while, but not anymore. So now we've got a few key messages, like Filipa was telling us about, of what we can do to sort of improve your systems. And that is, of course, you take into account your crop diversity. You want to diversify crops, but you want to have the right crop planted at the right time. If it's an annual, obviously for vineyards, that's a bit more tricky. You want to reduce mobilization, because soil is a habitat, and every time you plow, you're sort of destroying all this connectivity and habitat complexity of all these organisms. Keep it covered and enrich it in organic matter. So if you click again, that's all very well, but you want to quantify outcomes, because I can come to the farmer and say, oh, you need to diversify crops and reduce your tillage and keep the soil covered. And they're going to say, okay, but how many crops then do I have to have? What crops do I need to have? When can I plow, or do I just not plow at all? So we need to have some sort of guidance of what we're doing to this system and how it's going to respond. And I've got a couple of examples here of what we've been doing. So if you click again. Yeah, so two very different research. This one was actually a master's degree by our student João Souza. And he was looking at the Barroso ecosystem. The Barroso in Montalegre, in the north of Portugal, is our only globally important agricultural heritage system, according to the FAO, the Food and Agricultural Organization of the United Nations. So it's something that's being, very researched upon, very documented, and very, everybody agrees that this is a very sustainable system. And it's all about all these cows. This is beef cows that are free ranging in these permanent pastures, and then there's little crop fields and houses and the natural environment surrounding them. And there's lots of biomass and energy transfer between these three natural pasture and agricultural systems. So we went and sampled all of them. So if you click again, and we tried to see what was happening in terms of the soil complexity and in nutrient enrichment and functions as inferred through nematode community analysis. And you can see here on the left-hand plot that all of those agricultural natural pasture sites were all highly structured. So there's lots of food web complexity, lots of natural regulation there, stability for all of them, which was, well, I was going to say it was unexpected, but obviously this being a very sustainable system, this above ground sustainability is being matched below ground as well. And if you look on the right, then you look at the orange line is for your agriculture sites. And in agriculture, you have a lot more herbivory. This is all a logarithmic scale. So if you have just a 10 point there, it's actually times 10. So you've got a lot more herbivory in the agricultural system, and that's expected, yes. Fairly good regulation across systems, and a lot of nutrient recycling. That's probably being improved by the beef cows, really, having the animals there. So, and again, a very different system. If you click again, Rita, a very different system that we then, that I brought to you is nothing to do with sustainability, really, or hopefully we can make it more sustainable. And that's protected cultivation of tomato. So fresh market tomato in greenhouses across the country that I've sampled some years ago now. And we looked at all the plant parasitic nematodes that they had. And you can see those circles there are just proportions of different genera of plant parasitic nematodes, so herbivores in this soil. And we were focusing on the right-hand side. You've got a root that's very deformed, that's got all these knots, and it's a heavily galled root by a specific root knot nematode, a specific genus of plant parasitic nematodes. And we were worried about that one because it can be very damaging to agriculture, and it can in this photo up on the right-hand upside, you can see a greenhouse that they lost everything. They didn't harvest a single tomato from there, so 100% crop loss. And if you look at the faunal profile on the left, then you see that in different management, so large farms or small family farms or organic farms, they all end up, all those communities all end up in the very degraded area quadrant of the graph. So no structure, no regulation, and too much nutrient enrichment. So I went and sampled, but of course I talked to the farmers and I wanted to know more, and I had local observations and analyzed soil for other parameters as well. And I talked a lot to the farmers to see how they manage, what kind of practices they used. So if you click again, Rita, we come up with a very busy slide. And this is a canonical correspondence analysis. So what you've got, the words there in blue are plant parasitic nematode genera. And outlined in yellow, you've got different practices, you've got different properties of soil. You've got SFP and EFP. These are enrichment and structure footprints calculated from the nematodes. And then on the right-hand side, just to break it down a bit, so looking at the x-axis on the right-hand side, you've got enrichment, you've got chemicals, you've got resistant cultivars and phosphate. Yeah, okay, this is the last slide, she says. And then on the left-hand side, you've got structure and organic matter, rotation and intercropping, and application of biopreparations that the farmers come up with. Of course, these are very contrasting situations. And if you click again, on the right-hand side is where you've got your root knot nematodes, Meloidogyne there, and you can see that these different farms with different management, so organic or conventional small and large farms, really end up with very different nematode communities and very different plant parasitic nematode pressure and herbivory. So if you click again, this is the last slide. So we've been applying nematode community analysis to all sorts of different environments, not just agriculture. You can use them for any type of soil, really. And if you click again for the animation, then just wanted to tell you very briefly about this project, Soil Recon, in which we've been doing the nematode community analysis throughout the full north of Portugal in many different land uses, and we're coming up with very interesting data on land use type and intensity and all sorts of different parameters affecting as drivers or pressures to soil functions in the north. So next slide, and that's the final slide, just to say thank you very much for your attention and your patience, and I'm open for questions.
Thank you, Sofia. And now my challenge is we need to bring this map to the rest of Portugal. Why is the north the lucky one? We want to know the nematodes throughout as well throughout the territory. Sofia, thank you so much. Very interesting presentation. Sofia, we don't have much time, but I know that understanding the habitats of nematodes is essential for, ecological balance, agriculture, namely pest control, public health, environment management, pollution. But just to be more specific, how do you control for certain nematode communities? And also, you made it very clear that this is a very intricate network of relationships. So we always run into the problem of disturbing, the natural systems with consequences that we have seen throughout the planet. So how are they, how tangible? Can you tangibilize how you intervene in terms of nematodes? For example, you use the example of tomatoes. And what interventions did you recommend to those farmers?
Well, certainly looking at plant parasites, and not trying to control herbivory, you end up coming up with all sorts of suggestions, like Filipa said as well. These nematodes are just being used as bioindicators. Of course, we have a lot of work trying to figure out how to control plant parasitic nematodes that are in the soil. And those, we don't still have a silver bullet. And we need to come up with clever ways to make the ecosystem more resilient itself. Rather than targeting a specific pest or disease, we're trying to make it more regulated itself. And that's, again, having intercropping, strip cropping, ground cover of floristic ground cover. If you've got your permanent agriculture, you can't really change crops every season, but you can grow crops underneath your main crop, or you can just even allowing natural vegetation or sown vegetation to come in. That brings along all the life and complexity of the food web that you need to have both below ground and above ground. For nematode, for nematode problems, for plant parasitic nematode problems, you have cultural processes. You've got biopreparations that we've been looking into, that farmers have been using traditionally for ages, empirically. And now we're coming to the conclusion that some of them actually have a lot of effect. Solarization, biofumigation, whatever you do, you need to make sure that you're not using, or you use as few chemicals as you can. Even nematicides that you say they're only going to kill nematodes, they're still killing off all the omnivores and predators and all the nematodes that you want to keep. Yeah, and they have non-target effects throughout the full ecosystem. And so, yeah, organic matter. I could go on, Claudia.
Yes,
but
well, that's
usually
the challenge of managing it all and trying to identify where the balance and what are the most effective strategies. I can see that nematodes are at the forefront of science. We're trying to understand them a little bit better, although our grandfathers have been dealing with them for many, many centuries and successfully. And then this also, my question also relates to the question that we have here, is trying to understand how do we identify the nematodes. Are they easy to identify? I have the idea that they are hard to identify. So this participant says, in that matter, how easy it is to get the soil fingerprint using nematodes. So it's being practical about nematodes. Do we need to go to the lab? Do we need to do a genetic DNA sequencing as we do at our Arruda microbiome lab? Tell us a little bit more, Sofia.
Yeah, so in practice, what do we do? Well, we sit and look at them using a microscope. Are they easy to identify? For me, yes, but no, usually not. Usually it's easy to categorize them in trophic groups, but if you want to really come up with indices, there is a level of expertise involved for the classical identification and classical nematode analysis. Now, you can use method barcoding techniques. You can do the DNA. Usually, it's very fine. You get very fine taxonomical identification, whereas the indices that you use for all those fingerprints and footprints and indices, you need to categorize them into functional guilds, and sometimes it's difficult to do that with DNA. So we're still improving that part. But it is feasible, yes.
Okay,
how
great.
to avoid, sorry.
Sofia,
Yeah.
I need to stop you here, not to answer to these questions. So of course, I will ask you to use the chat, because otherwise I will risk Felix to leave, saying that Portuguese people are very rude and run very, very late. So Sofia, hope you understand. Just also to give the floor to Felix. Felix also has some very interesting things to share here with us, but I would like to leave our participants without your views on the question. So Sofia, please feel free to answer in the chat. And I now invite Felix to start sharing his presentation, and to let you know that Professor Felix Wackers has worked in the field of functional biodiversity for more than 30 years, including positions at Lancaster University, Wageningen, the ETH Zurich, and the USDARs. And since 2009, he holds the position of Director of R&D at the BiobestGroup, the world-leading company in the field of biological crop protection. Also, his academic research addressed the interactions between the crop and the semi-natural landscape, with special emphasis on optimizing biodiversity functions in terms of ecosystem service delivery, specifically pollination and conservation biological control. He has coordinated several large-scale projects on functional biodiversity, and he has led for 7 years the European Learning Network on Functional Biodiversity. Felix, it's a pleasure to have you here, and the floor is now yours.
Yes, thank you, Claudia, for the introduction, and also thank you for the opportunity to share my work on functional biodiversity here. We've heard some really interesting presentations already. I will continue with the type of work that Filipa was presenting, to modify or optimize the agricultural landscape in such a way that ecosystem services are delivered. I hope that my presentation will move forward here. This is unfortunate. Sorry, I have to
scroll down.
I'll try sharing again.
All right. Okay. So this is the webinar where presentations decide to have personal life. No worries.
Yes, I'm just going to. All right. Do you see the slides changing? Good.
Yes, yes.
So
thank you.
you're probably aware of the fact that we have seen quite dramatic declines in biodiversity over the last several decades. And there are a number of causes for this, climate change being one of them, but two main causes involve agriculture. And it's associated with intensification of agriculture. So as a result of intensification, you see that there is often a loss of landscape structures. You get these huge monocultures. And intensification also has led to an increased use of pesticides. And these two factors are two main drivers for the decline in insect biodiversity and biodiversity in general, because we have to remember that many other groups are dependent on insects as well as a food source. Think about birds, for instance. The decline that we see in agricultural bird species is also associated with the decline of insects. So early on already there were different national programs, agri-environment schemes, which tried to reverse this trend. And they were focusing on this negative impact that the agriculture has on the environment. Then farmers were given subsidies to introduce structures or do something to the environment to reverse this situation. And the focus of these agri-environment schemes, and especially in the UK, but also in other European countries, are often birds and butterflies, and in some cases pollinators as well. If you look at what happens, what do farmers do if you give them these subsidy options? In the UK, there are quite a broad range of options that farmers can choose from. And on the graph here, we see the number of hectares where these options are being chosen. So what you see on the left here, the big bars, those are low input grassland or mixed stocking, which are factors which have relatively little impact on biodiversity, so little ecological impact, but it's very easy for farmers to apply these. Whereas the ones here indicated by the arrows are the ones that have huge potential ecological impact, but farmers don't choose them because they are perceived as being complex. So we were thinking it would be actually good to turn this narrative around. So not focusing on the negative impact that agriculture has on the environment, but let's talk about the positive impact the environment has on sustainable farming, and thereby creating actually additional incentives for farmers to engage with agri-environment schemes. And when we're talking about these positive impacts, we are actually focusing on the story that biodiversity can work for farmers. So what are we talking about? Ecosystem services, that phrase was already mentioned before. And an important ecosystem service that people often think about when we talk about insects is pollination, which represents a large economic value. Constanza estimated this to be 90 billion per year on a global basis. It's believed that it's actually much more. But there's also another ecosystem service that we don't hear as much about, and that's biological pest control. So a lot of insects help the farmers in controlling their pest problems. And this, in the same study by Constanza, was actually estimated to be 3.5 times as important, economically speaking, as pollination. So it shows that it's a huge economic benefit that people usually don't pay a lot of attention to. Well, when we're talking about these groups of insects, the pollinators and the pest control agents, they both suffer a key bottleneck in modern agriculture, and that's the fact that these monocultures have a clear lack of floral resources. And we all know that pollinators, of course, need flowers to collect nectar and pollen. But what is far less known is that also insects used for, or that are relevant for natural pest control, are also dependent on flowers for energy, nectar, or protein, pollen. So we can actually address or stimulate these important group of beneficial insects by creating or reintroducing floral resources to the agricultural ecosystem. And that's something we have been working on for the last 30 years. Well, to show you that this is not just a trivial matter, here's some study that was done by my PhD student in Wageningen, where she worked in field cages with cabbage plants. In these cabbage plant cages, she introduced a single parasitic wasp of the cabbage moth, Prutella. And then in half of the cages, she added one flower of the buckwheat plant, and the other half of the cages were kept without the flowers. Then the pest moth larvae, so the caterpillars, were introduced every day, and then at the end of the day were removed, and we looked to see if these caterpillars were attacked by the parasitic wasp, if the parasitic wasp had laid an egg in the caterpillars. So what you see at the bottom here is the cages where no nectar plant was introduced, and we see that the wasps lived a maximum of 6 days. And in these 6 days, they managed to just parasitize a handful of caterpillars, so very few. In sharp contrast, all the cages which contained buckwheat flower, the parasitic wasps lived 25 to 35 days and were able to attack and parasitize more than 300 caterpillars. So we can see that simply by introducing a floral resource in this system, you can have a huge impact on natural pest control. And then you may say, okay, you've chosen these parasitic wasps. That's maybe just an example where it's very clear cut. But here is an overview that we created for a book on this topic, where we were able to show that all the groups of insects that are relevant in natural pest control, so all predators and parasitic wasps, are also dependent on nectar and pollen feeding. So really, by not having these floral resources in our agricultural system, we doom these biocontrol agents to starve and to be ineffective. So if we want to reverse this, if we want to actually support these beneficial insects, how do we go about this? How do we optimize ecosystem services? Well, the one point that I would like you to remember from this talk is that we're talking about different insect species, and these have different flower requirements. So if we look at creating floral resources, we should not be thinking that we can have one size that fits all, but we really have to take a tailor-made approach where we look at what are the nutritional requirements of these groups, which flowers species are being visited and exploited, and so that we can tailor the flower composition to the groups that we want to support. To show you how this works, the type of work that we have done over the years, here we see a group of flowering species that are categorized according to the depth of the flower. So how deep in the flower is the nectar to be found? And we already heard from Filipa that different insects have different types of mouth parts. Well, the pollinators like the bees and bumblebees, they typically have very long mouth parts, so they can get to nectar that is very deep inside the flower. But if we look at insects relevant for natural pest control, they have very short mouth parts, and they are dependent on very open and accessible nectaries. And that's what we see here, especially the parasitic wasp. They only accept flowers that are where the nectar is completely open. In the case of the hoverflies, they can accept flowers where the nectar is 2 millimeters deep, and about 3.5 millimeters is acceptable for the lacewings. So if we want to then select flowering plants for biocontrol, we take the flowering plants here in the top that are suitable for all groups of biocontrol agents. Well, this was all based on kind of lab and semi-field studies. On the basis of this information that we gathered, we actually also started doing large-scale field studies. And a good example of this is the functional agrobiodiversity study in the Hoekse Waard, which is south of Rotterdam in the Netherlands. We initiated this project in 2005, and so it has been going now for 20 years. After we ran the project for 6 years, the farmers were so enthusiastic that they actually started a cooperative to continue this work. And by now we are having here more than 800 kilometers of flower margins. And there are more than 200 farmers that are involved. And there's a waiting list for farmers to join this project. In addition to this project in the Netherlands, I've also been involved in projects in the UK when I was at Lancaster University working in vegetable crops and in orchard systems. I've now been involved in projects in Germany where the focus is on sugar beets, and we are doing work in Belgium, and in Switzerland I worked on grapevines. And there's 2 projects in the pipeline, and one going to be in Portugal, hopefully, and the other one going to be in Sicily. So we're also expanding this approach to the Mediterranean cropping systems. And I was very happy to hear of Filipa's presentation, to see all the interesting work that's going on in Portugal already. So just to show you some results of these trials here, some work from the UK where imagine you have a field, which is the white one here. On the one hand, you have a flower margin. On the other hand, you have other side, you have a grass margin. And then you can see what kind of insects do you find on the flower margin, which is blue here, and what on the grass margin. Well, you see that the flower margin clearly stimulates the insect visitation, which is not very surprising, and that this effect actually increases over the years. So from 2010 to 2011, the effect is clearly more pronounced. What is more interesting in terms of convincing farmers is to look at what is happening in the crop. So here again, we results from a pea crop. On the left, the flower margin, on the right, the grass margin. We looked at the pea aphids here as an important pest. And the blue bars show you the population development of the aphids over time. So this group here next to the flower margin, this group next in the center of the crop, and this next to the grass margin. Well, you see that near the grass margin, which is about 150 meters away from the flower margin, the aphids are actually increasing throughout the season, and at the end of the season, the aphid population was actually too high for the farmer to accept. Next to the flower margin, and also in the center of the field, we see that the aphid population doesn't increase, it actually decreases over time. And the explanation for this is shown in the black line, and that's the parasitism rate, so the number of aphids that were being killed by parasitic wasps. And you see that these parasitic wasps next to the flower margin occur much earlier in the season and in much higher numbers. And that explains this improved natural pest control. So to summarize what we've seen in all these different projects working with often conventional farmers, is that we can achieve reductions in pesticide use by, in the case of the Netherlands, by more than 90% in wheat and in potatoes. And we also, which is very interesting, see an increase in yield in a number of these studies. So in the UK, we saw, for instance, an 11% increase in the wheat and 26 in the peas. In the Netherlands, we've seen 30% increase in beans, where in the case of beans, it's probably due to both biocontrol and the pollinators that are being stimulated. So this shows you that this approach, and I know the discussion before went into the direction cost-benefit analysis. We see that sustainable systems and productivity can go hand in hand. They don't need to be conflicting objectives. So we create, by having the right type of flower margin, you can create a positive spiral where you stimulate your beneficials. The beneficials move into the crop and suppress the pests, which means fewer pesticides need to be used, which in itself is another supportive mechanism towards supporting beneficials and supporting biodiversity in general. And yes, I think my time is probably up as well. And I thank you for your attention. If there's still time for a question, I'd be very happy to answer those.
Okay, Felix, thank you so much also for another very interesting presentation. Very interesting, and you know, the trade-offs and the cost benefits are something that are really, really important and always on the top of you know, the farmer's mind, also mine, because I have a business background. So thanks for addressing that. And I think this question here in the chat also address that. So so we have the evidence for the figures, okay? You have shown that this can work. So how can we gather more interest from farmers towards these approaches and to work with the functional biodiversity?
How we can, well, my experience, and let's see, yeah, my experience with working with farmers is if you can include them from the beginning of the project and have them have their say what their concerns are, what they hope to get out of this project, but also what their concerns are. And if you listen to farmers, often they have a concern about when you use flower margins that you may stimulate weeds or that you may stimulate pests. And then we can explain to them how we select these flowering plants, that we only use plants that don't create any weed problems, that we also have done studies, which I haven't been able to show you today, but that you can select flowering plants that are beneficial or provide food for the beneficial insects, but not for the pests. So there is this tailor-made approach can be used to exclude benefits to pest insects as well. And that way farmers see that their concerns are being listened to and that they're being taken serious. Also, what we often see is that the farmers initially might be interested, but might also be kind of skeptical about the approach. But when you do your monitoring and you do your scouting and you can share these results with them, you can take them to the field and especially these flower margins are really suitable for that. You can take them to the flower margin. You see the hoverflies and the ladybirds and the lacewings on the flower margin. Then you go to the field and you see the larvae of the ladybirds feeding on the aphid colonies. And you see the larvae of the hoverflies. So it's very clear. You can visualize how these flower margins work. And by doing it this way, you farmers usually become very enthusiastic about these kind of approaches.
Yes, giving more voice also to those that already tested and perceive the benefits of working with these flower margins. I have a final question, and I want to keep you here for all of you. But for Felix, it was very clear from your presentation and from Filipa's presentation that we need to be careful at selecting the species and the insects and the pollinators that we're going to introduce, because not all of them are beneficial, or at least are not as efficient to target our pests. My question is, because we know that a lot of species face extinction, some of them have completely disappeared. So how confident are we still that we can still depend on the natural environment to attract these species? And if they're not available anymore, is it possible to raise them in the laboratory or to get them somehow? Or you don't feel that this is a problem yet? We're not in that such severe situation.
Well, if I may answer, I'm sure the others have something to say as well. Yes, you can. We are insects for biocontrol in companies. My company does this, but I want to make sure that this approach is conservation biocontrol. So it's actually something my company lets me do this because it's my academic work, but a company like BiobestGroup is not going to make any money off of this approach. It's in a way you could see it as a competition. I think that most of the biocontrol agents are still present in the larger landscape, but often not in sufficient numbers, and often they don't have the right
environment
resources available to build up numbers. So what we see in our projects is that if you do this, and especially if you do it also on a larger scale, then you can build up these populations over 1-2 years. So it's fairly quickly. But if you do also need some time to build up these populations. So the effectiveness of these approaches is better if you have more time to keep these flower margins. And that is something we always recommend, to have perennial margins and perennial hedges, because those allow not only food sources for the beneficials, but also overwintering sites.
Okay, good. That comes as a relief to know that we still have animals to save us, and we haven't. But I would say that time is not on our side. Filipa, do you agree?
Sim, eu concordo. Dos trabalhos que nós fazemos, verificamos isso mesmo. Há insetos que os números são reduzidos, bastante reduzidos. Mas fora das áreas agrícolas conseguimos ainda encontrar populações, e há de facto insetos que podem ser reproduzidos em laboratório, mas que, pelo menos aqui em Portugal, o que eu verifico é que essas largadas de insetos, de auxiliares, são feitas já como se fosse um tratamento e não como uma reposição de insetos que depois deveriam fazer os seus ciclos naturalmente na natureza. Portanto, estão a recorrer como se fosse um tratamento, e o objetivo, de facto, acho que não seria esse. Isso seria para recolonizar em sítios onde as populações estão, de facto, muito diminutas, mas sempre na tentativa de os estabelecer na exploração e não como uma rotina. Agora, eu acho que o importante para aumentar essas populações passa muito pelas práticas agrícolas, não é? Nós não podemos estar a colocar sebes e a investir em cobertos vegetais e a aumentar as riquezas florísticas e depois não considerar isso quando pretendemos fazer tratamentos. Portanto, aqui temos que dar tempo à natureza para poder dar essas respostas, porque estamos a gastar dinheiro de um lado, estamos a investir de um lado e depois estamos novamente a gastar do outro quando fazemos tratamentos sem esperar para ver se realmente as coisas estão a funcionar ou não. Mas também há aqui outra situação que eu acho que é importante, que é o desconhecimento, não é? Portanto, nós estamos a falar de insetos que não se veem, e muitos dos produtores nem sequer sabem que insetos é que são os mais aptos. E, portanto, a falta de conhecimento e a falta da chamada extensão rural, ou seja, que dá este aporte de conhecimento aos agricultores, é importante para que eles, de facto, tenham também a sensibilidade de ajustar a gestão deles, não é? Porque, numa dúvida, naquilo que não se vê, a primeira coisa a fazer é um tratamento. E, portanto, estas questões ainda têm que ser muito ainda discutidas e faladas, sim.
Sem dúvida, Filipa, obrigada por alertar para esses pontos importantes. Quer dizer, isto precisa de tempo para se construírem estes recursos, não é? Que o Felix também estava a falar. Se me permitirem, só mais um bocadinho do vosso tempo, if you allow me just a tiny little bit more of your time, I would like to know your opinions about the new PAC. So the new PAC has strongly advertised that they are going to invest or to incentivize farmers on biodiversity. And we assume that it's, of course, also biodiversity, functional biodiversity. What is your views on how things have been working? Are we short on that? Are we achieving, at least starting to walk the talk? what is your views in terms of the assistance of the new PAC and biodiversity? And this is for all of you. I know PAC is a hot topic, but it would be interesting as experts on biodiversity to hear your views. Would like to start? That was a tough question.
No, I can say something about it, but I don't want to be the first necessarily to say something. But yeah, I think that it's very important, obviously, to reintroduce and to reestablish some of the biodiversity in the agricultural landscape. Agricultural landscape is the most predominant use of land use in Western and Southern Europe. If we impoverish this landscape by having larger and larger monocultures, then we really see the impact that it has on biodiversity in general. So all kind of biodiversity groups are suffering, and especially also the functional biodiversity. And I think it's important for farmers to understand and to be given information that shows them that these biodiversity measures that are being supported, that it's not just something that is helping environmentalists, but it's also something that is really essential to sustainable farming and farming productivity. And if we are able to bring this message across, farmers will actually be actively involved and will become champions of this approach. And that's what you were saying before, Claudia. It's good for researchers to be presenting these examples. It's even better if farmers can share these examples among each other, and that way show that they have the first-hand experience that this works and that it's good for them. And that way we create a win-win for farming and the environment.
Yes, definitely. Farmers need to be confident about these measures, so we need to show more evidence. Filipa, PAC and functional biodiversity.
É assim, claro, é uma ajuda, não é? É uma ajuda, e até porque o caminho não é o outro. Eu estou mais da parte dos agricultores, portanto tenho um contacto se calhar muito mais direto com eles, e de facto nós estamos muito condicionados, e por isso o caminho tem que ser pela biodiversidade. É a base do sistema, é por ali que nós vamos conseguir o equilíbrio que tanto desejamos e que no fundo nos vai dar um suporte para a nossa produção. A falta os condicionamentos que nós temos relativamente aos tratamentos, aos produtos fitossanitários, às substâncias ativas disponíveis, é um problema, e a longo prazo esse problema vai permanecer e vai se intensificar. Portanto, sempre que se comece mais cedo a investir e a trabalhar com a natureza, automaticamente o input dos fatores de produção vai crescer, portanto a nossa dependência deles vai ficar menor, e isso é uma mais-valia, não é? A resiliência depois não vem só na parte cultural, vem também na parte económica, porque a sustentabilidade tem aqui 3 pilares muito importantes e não podemos olhar só para um, nós temos que olhar sempre para os 3. O económico é importante e tem que lá estar, o social, mas também o ambiental. E, portanto, a questão da biodiversidade e a PAC vir frisar essas questões é determinante para mostrar que o caminho é de facto esse, já não dá para voltar para trás.
E mais remuneração também aqui dos serviços do ecossistema providenciados, não é? Pela esta biodiversidade funcional, algo que está, se calhar, lagging behind, mas temos esperança que isto seja aqui o princípio de uma revolução para, de facto, utilizarmos mais a natureza a nosso favor e a favor dos sistemas produtivos, porque a agricultura é, acima de tudo, um sistema produtivo. Obrigada, Filipa. Mas antes de ir, Sofia, has PAC reached the nematodes? Or are we still far away from nematodes?
I think we really need to transform the way we do agriculture. We have changed it completely in the 70s, and now people have become somehow hooked to all these inputs and reactive form of doing agriculture. We need to really bring back biodiversity and functional biodiversity into the system. If the new pack is going to help, we'll see. But we really need to start thinking differently and investing not in high yields, but in stable yields, that even if there's adverse years and adverse weather and pests and diseases that are emergent and we hadn't had them before, we need to be able to face all that and keep some sustainability and stability in the yield. Now, nematodes are never represented anywhere. We've got, thousands of species. None of them is in the IUCN Red List, but we want the functional biodiversity of nematodes in the soil and the full ecosystem working for us. So, yes, we need deep changes in the way we do agriculture and biodiversity. Definitely, we've learned so much. We need to apply that now.
Yes, thank you, Sofia. It was great. And we have here a great audience, and we are all together to promote this change. We are just kicking off the debate. I have to call to an end this webinar just to let you know it was a real pleasure to moderate and to hear all these great presentations. I hope that you have enjoyed. Hope that you have other questions and more food for thought. Remind you that we'll be back next week at the same time, looking at tools and indicators of functional biodiversity. And I hope you can join us next week. Once again, many thanks for your support. Many thanks for your time. See you soon. Bye.